Bismillahirrahmanirrahim. Assalamu alaikum. I'm Juwaria Kadus along with Khola Tariq from Fatma Jinnah Women University. Today we'll see the titrations in determination of food properties. In colleges, almost all science students learn about titrations. It's not just a chemical experiment that we do in colleges, but it has a wide range of applications in different industries and in different aspects of our daily life. Today we will see the applications of all the four types of titration in food sector. So let's start with applications of acid base titration. Food acidity is due to the presence of organic acids like malic, lactic, tartaric and acetic acids, and the presence of inorganic acids like phosphoric and carbonic acids also play a predominant role in food acidity. So we can determine the food acidity by acid base titration by neutralizing the acid present in a known quantity of food sample using a standard base. Normal pH of milk ranges from 6.4 to 6.8, making it slightly acidic or close to the neutral. So this acidity of the milk is mainly due to the presence of lactic acid, which is produced by the action of bacteria lactase on lactose in the milk. Let's see the procedure for determination of acidity of dairy products. So in the first step, 10 normal solution of NaOH is taken in the burette and the sample of dairy products is diluted with distilled water. Then 3 to 4 drops of phenolphthalein are used, an indicator which is an acid base indicator, and color as in the acidic medium, whereas pink in the basic medium. Then titration will be done till the pink color endpoint and the final burette reading will be noted. So this is the formula to calculate the percent lactic acid or percent acidity in the dairy product sample, and the weight of the sample is a product of volume and density of the sample. So through this procedure we can calculate the acidity of different dairy product samples like milk, cheese, yogurt, butter and ice cream. So next is determination of acidity in vinegar. So acidity in vinegar is mainly due to the acetic acid, which is derived by the fermentation of ethanol by acetic acid bacteria. So we can neutralize this acetic acid with by titrating it with a strong base. So in the procedure, a sample of the vinegar is titrated with NaOH till the phenolphthalein pink endpoint, and then the reading is noted. So in the calculation, the following formula is used to calculate the acidity of vinegar, where the weight of the sample is the product of the density and volume. The acidity of natural fruit juices is a result mainly of their organic acid content. For example, orange juice and the lemon juice are slightly acidic due to their citric acid content. In the procedure, a sample of fruit juice is taken and is titrated with sodium hydroxide till phenolphthalein pink endpoint, and the final reading is noted. So this is the formula for calculation of percent acidity due to citric acid in fruit juices, and we can see a 3 in denominator because through a balanced chemical equation we can see that the 3 moles of sodium hydroxide are required to neutralize 1 mole of citric acid. So next is the determination of wine acidity. So organic acids in wine are primarily derived from the grapes. However, due to the alcoholic fermentation, some major acids, for example acetic, lactic and succinic acids, are also produced. So this acidic content of the wine is determined by titrating a sample of wine with a base sodium hydroxide to the phenolphthalein pink endpoint, and the acidity is then expressed as grams of tartaric acid per 100. Next is the determination of oil acid value. So the acid number, the acid value is defined as the number of milligrams of potassium hydroxide that are required to neutralize the free fatty acids present in 1 gram of fat or oil. So this is a relative measure of density of oil by estimating the free fatty acids content produced by the fat fermentation. So in the procedure, a sample of oil that is dissolved in water an ethanol mixture is taken and is titrated with KOH solution in the burette till the phenolphthalein pink endpoint and the final reading is noted. So in calculation we can say its formula, it is not a percentage but an acid value, so it is not multiplied by 100, and this factor is the molar mass of KOH, and the weight of sample is the product of density and volume. Next comes the application of another type of titration, which is redox titration, and we know that redox titration involves the oxidation reduction reactions. Vitamin C in food samples can be measured through redox cytometric titrations. So through this titration we can calculate the vitamin C content of vitamin C tablets, fruit juices, and the solid fruit and vegetables. So in the procedure, iodine is used as a titrant which is taken in the burette, and the food sample along with starch, which is used as an indicator, is taken in a flask in which ascorbic acid is an analyte. So iodine oxidizes the vitamin C, and when all the vitamin C or ascorbic acid is oxidized, iodine reacts with the starch and the solution turns blue-black, which is the endpoint. We can measure the amount of iron in different food samples through a redox titration called permagnetometric titration, in which potassium permanganate is used as a titrant which is taken in burette, and the food sample is taken in flask in which iron is the analyte. So as we know that potassium permanganate has a bright purple color, so it acts as a self indicator on reaching the endpoint. We can also calculate the iron content of the food samples through dichromatometry, in which potassium dichromate is used as a titrant taken in the burette, and the mentioned indicators are used, and the color change of the indicator is from green to violet.
Assalamu alaikum. I'm Khala Tarek, here to talk about the applications of precipitation and complexometric titration in food. So first of all, talking about the reducing sugars, they can be determined with the help of Felling's test, which come under the precipitation titration. It's basically used to determine water-soluble carbohydrate and ketones, while a test for reducing and non-reducing sugars. You can see its formula, that is cupric sulfate. This reaction, it requires basic medium. In this type of reaction, there is a precipitation of copper. So to avoid that, we can add tartrate ions. So the procedure includes two steps. First is the standardization, in which this freshly prepared Felling solution is titrated against glucose to form standardized Felling solution, and after that the standardized Felling solution is titrated directly with the corresponding reducing sugar that we want to test for. You can see the reaction in which a sugar is converted into its respective acid, while the blue color solution of Felling, it is converted into its red precipitate, that is the end. So talking about the applications of complexometric titration in food, the main application of this titration is the phytic acid, which are found in different foods like juices, milk, and cereals. I'll be discussing them one by one in the next few minutes. Talking about the phytic acid, it is found in plant-based foods and beverages. It can be measured through a complexometric titration by titrating against ferric ions and EDTA, that is ethylene diamine tetraacetic acid. You can see the formula for it, which is a very complex. The procedure for the determination of phytic acid in juices and milk includes many steps. First of all, take a test tube and to it add HCl, iron-3 solution, and 20% of salicylic acid solution. And after that, place the test tube in a hot water bath for about 15 minutes. When the solution in the test tube is hot enough, then centrifuge it for about 10 minutes. After the completion of centrifugation, take a little amount of the supernatant solution and add it to the flask, and to which dilute the solution with water, and to maintain the pH at around 2.5, add a small amount of glycine and then heat the mixture in the flask. When the flask mixture is heated enough for up to 60 to 70 degrees centigrade, then it's the right temperature to titrate the solution with EDTA. which will induce an endpoint. The purple color of the solution previously that was a complex of iron and sulfosalicylic acid, it will change to a bright yellow color that is a complex of iron and EDTA complex. That was the procedure that we discussed for the determination of phytic acid in juices and milks. Now we will discuss its determination in cereals. Basically, phytic acid is also found in many of the cereals. It can include flours, milk, wheat products, brawns, breads, etc. And in some brawns, it can contain over 5% of phytic acid. So we'll proceed towards its procedure. The initial steps for the determination of phytic acid in cereals are same to those which we discussed for the phytic acid in juices and milks. It includes the same preparation of a solution of phytic acid which we heated in a water bath and then that was centrifuged. So take a small amount of that prepared supernatant solution of phytic acid in a flask. To that flask add glycine, which is to maintain the pH from 2.5 to 3. Add sodium sulfate, HCl, dilute the solution with water, and after that add the flour of that particular cereal that you have to determine the phytic acid in it. After that, heat the solution that is present in the flask, and when the temperature is appropriate, titrate the solution against EDTA. It will mark an endpoint, and the complex formation will convert the solution color, that is red maroon, to. So that was all the applications of the titration among the food category. Hope you understand all of the points that we have discussed up until now. Thank you so much.